Afternoon.
My name is Madam Magoy. I will be taking you for microbiology class, and it's a topic that is so dear to me, and I believe that by the end of the lesson, you have taken something that will help you even in the future. So today, what's the date today? That 10, 20, 24. So our first topic, we start with a test that is called vital staining. That is developed to give us to differentiate the living and the dead cells. Once you get microorganism in any product, it is very, very important to carry out this test to know whether you're dealing with living organisms or dead cells. So in this kind of technique, the principle that is usually used is methyl blue, that will ensure that the dead cells will take in the blue color and the living cells will not take in the blue color. So in this test, there are apparatus that are usually used. Give me an example of the apparatus and reagents used in that procedure. Microscope. Okay.
We have the slides that you put on the microscope.
Yes.
Beaker.
So I'll read for you some of these apparatus that are used. We have methyl blue solution. We have the microscope, as you have said. We have grass rights. There is an inoculating loop, a staining bottle, hoping you're with me in writing. We have staining lag. We have distilled water, capacity, and the Bunsen burner. Each and every apparatus or equipment that we have mentioned have a specific role, and that is now your assignment. You go write for me all the purpose or the uses of whatever I've mentioned. You go now to the procedure. Maybe you can make a guess of the procedure that is used in carrying out the vital technique. Who can try? Okay, you start with, you take the sample with an inoculating, you place it on a grass, right? Yes?
I think we should sterilize
In this case, it's a procedure. Once you go to the practical, yeah, you should ensure that all those processes are followed, right? So I'm just giving you the procedure, right? So that when you're going to carry out your experiment, you know all that is supposed to be done, right, class?
Yes.
Any questions so far? So the procedure is you take the grass light and you clean it well. As he was saying, most of the lab apparatus are sterilized. But once you use, you're supposed to sterilize again. Yeah? So you take an inoculating loop, you place sample on the glass light. Then you add methyl blue. Once you put the methyl blue, you put the cover slip. Then you remove the excess dye with what distilled water. Why do you remove the excess dye? I can maybe try, shall we?
To remove the excess dye.
Yeah.
We remove the excess dye for proper or final, so that you can see and observe clear images.
That is very, very correct. Okay. So when carrying out that laboratory test, there are some meters that you should take or you should know at the back of your head. One is, what are the risks when you leave the Bunsen burner on and you're not using it, there is a risk of fire. Okay? So from there, from the previous class that you have been doing, I would like you to continue now from there. You give me the procedure, the complete procedure of staining method technique. Have a lovely afternoon.